And I would like to give a couple of words about the chemoinformatics, bioinformatics, rational drug design, what we are doing so far. And this is the group. We are collaborating with a lot of people, students, professors from the US, also Professor Kravtsov. He was already presented some data, and he's actually very actively involved in our project. So we might do some projects together if you are willing to participate in our various projects. So just a couple of slides to give you some ideas what we are doing so far. So as you can see, we have some projects concerning food informatics, bioinformatics, where we have some proteins, and these proteins might interact with some molecules, the food molecules, and some food molecules are extremely important, for instance, in terms of the ability to stimulate the receptors in the body. For instance, this is the artificial tongue bioreceptor, biosensor as we call it, and we are creating some kind of a hybrid system based on the transporter, the receptor, and this receptor, the protein receptor, interacts with different sweet ligands. So after that, we put this system put the system to the analysis, and we have to process the binding affinities and how these binding affinities are changing using different ligands. The other is chemoinformatics and triboinformatics with Professor Nosanovsky. And here we are trying to evaluate how the different proteins or small proteins, small kind of peptides, they will fold using some parameters. And we are trying to develop new algorithms or new functions. Here we are using some kind of alpha, which will give us the idea and how these proteins will go from unfolded states to the folded states, and for the different metastates. And this is particularly important in terms of the diseases, in terms of the structure of the proteins, and how we can probably, if you mutate protein, we can predict probably the structure of the proteins and create a better medication or the better biosensors. Again, this is the other algorithm, and this algorithm is specifically important to analyze the folding, unfolding, and symmetry of the protein. If you can see, we have the unfolded state and the folded state, and there is some kind of a symmetry, and we can actually predict the folded state from the unfolded state, and vice versa, using the molecular dynamics. Here I'm not going into details, but this process, the energetically dependent, and as you can see, the process itself is different in terms of the time. If you're going from unfolded state to the folded state, usually you need more time, and vice versa, it will be extremely fast. We're also doing on the software design, and here's the software, which is called NeuroClick, and this NeuroClick algorithm is giving us the idea, the ligand using the click chemistry. And by using this click chemistry algorithm, you can generate the database of different drug-like molecules, and after that, these drug-like molecules can be further evaluated by using the blood-brain barrier permeation function, and one could predict the better medication by understanding how the substance would permeate through the blood-brain barrier, which is extremely important to treat some neurological diseases, for example. The other project is chemoinformatics and computational pharmacology, where we are doing the formulation of different substances in order to improve the solubility of the substance, or pharmacokinetics and pharmacodynamics of different substances. Here, for example, the excipient, different excipients. One is the hydroxybutyl beta cyclodextrin, and the other is sulfobutyl beta cyclodextrin. And now we're trying to understand how the substitution degrees of this excipient molecule would increase or decrease the binding affinity, hence improving pharmacokinetics of different substances. And last but not least, chemoinformatics and ergodic therapy of cancer. This is the new paradigm in a modern precision medicine, where we are trying to understand and use this molecular mechanism, which is called chromatopsis, to treat some diseases such as cancer. And we are trying to investigate how the mutation would occur if we will stimulate the cells to go through a particular threshold, chromatoptic threshold, and how these mutations would affect the protein structures. First, they would affect the genes, and then if the gene is expressed, you would have some mutations within the protein structure, and how this would actually deal with the stability of this particular protein. This is BCL2 protein, which is extremely important for the cell survival and replication. And of course, this new paradigm would give us some benefits against this very deadly disease. Okay, thank you for your attention. This is all about it. If you have any questions, I would be happy to answer. And I'm waiting for you to embark on this exciting projects to have a Nobel Prize in the future.